Welcome to Synapse City, where every nerve signal is like a message that is zipping through a busy street. But when communication piles up, chaos starts to break out. That's when our biochemical hero, ACE, which is short for acetylcholine esterase, takes action. Scientists call him HE, but we'll call him ACE in this story. What is his job? Well, it's to keep the neural highways clear by breaking down extra acetylcholine after each signal. This superhero story is a lot like what happens in our nervous system. Every time a nerve fires, acetylcholine molecules flood the synapse, which is the tiny gap between the two nerve cells. ACE, which is an enzyme, will catch these molecules and then split them up into acetate and choline. This quick cleanup helps reset the synapse and lets communication flow smoothly. But like with any system that is under pressure, there's always a limit to how fast ACE can work, and that limit is what the Michaelis-Menten model helps us understand. Imagine measuring how fast ACE breaks down acetylcholine as you add more molecules. At first, the rate goes up sharply because there's a lot of room for each molecule to attach to the enzyme. But soon, as the concentration grows, every ACE molecule is occupied, and adding more acetylcholine won't make things go any faster. This is where the reaction plateaus at its maximum rate, which is called the Vmax. In the middle of this curve is Km, which is the Michaelis constant. Km tells us how strongly the enzyme will bind to its substrate, so in this case, how strongly ACE binds to acetylcholine. We can find Km by looking at the substrate concentration where the reaction speed is at half of Vmax. This point on the graph shows us how easily the enzyme can reach half of its top speed. If the Km value is low, then ACE is really good at grabbing acetylcholine. If the Km value is high, ACE needs more acetylcholine to get up to speed. Collectively, both Vmax and Km show us how efficient the enzyme works and the enzyme's capacity. Scientists use the same curve to understand how enzymes perform under pressure and where their efficiency peaks. These patterns aren't just theoretical. Rather, they explain how our bodies stay balanced. Enzyme kinetics influences how our muscles contract, how we digest food, and how we respond to medicine. When enzymes like ACE work too slowly, then signals start to linger and muscles will misfire. When they're too fast, or when they're blocked, things can get out of control and communication shuts down. So understanding these limits helps keep every system, from our smallest synapse to even our largest network system, to work in harmony with one another. Even heroes face villains. In Synapse City, imposter molecules, which are the competitors that mimic acetylcholine, will sneak in and race for ACE's active sites. These are competitive inhibitors. Because they resemble the real substrates, ACE sometimes binds to these imposters by mistake. This means that the reaction will slow down at first, but if we keep adding more acetylcholine, then those real substrates will outcompete the imposters, and eventually ACE can still reach the same maximum velocity, or Vmax, as before. However, the concentration needed to get there is now higher, so the Km shifts to the right on the graph. Therefore, the Vmax stays the same, but it takes a higher concentration to reach it, which means the Km increases. Other enemies will attack differently. These enemies will attach onto ACE's side, which changes his shape. Since ACE has a different shape now, he can't function properly. These are non-competitive inhibitors. These molecules don't directly block the active site, but rather they attach onto the allosteric site, which changes the shape of ACE. Now, even if there are a lot of acetylcholines flooding into the synapse, they still can't attach on effectively. Therefore, the overall Vmax decreases because there are fewer enzymes that can work. But since the enzyme's ability to recognize the substrate hasn't changed, the Km still remains unchanged. In the end, ACE shows us that even the most efficient systems have limits as well. The Michaelis-Menten model helps us understand those limits. Specifically, it shows us how enzymes reach their peaks, how inhibitors challenge them, and how balance keeps life going. Enzymes like ACE are the unsung heroes that are behind every heartbeat, thought, and muscle movement that make sure our biological cities run smoothly.